Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. In this video we are going to discuss the thymus gland and the development of T cells. First of all, thymus gland. It is a primary lymphoid organ. It develops during third month of intrauterine life. From the third or fourth pharyngeal pouch. It is highly active at the time of birth, reaches its peak action at the time of puberty, and then gradually it degenerates. Now, this is the structure of thymus. Thymus is bilobed in human and is located anterosuperiorly to heart just beneath the sternum. These are the two lobes. This one is left lobe and this one is right lobe. The upper or the outermost covering is known as capsule, and these are the interlobular septa, and this is cortex, and the innermost region is medulla. Now, thymus is divisible into cortex and medulla. Cortex is densely packed, while medulla is less compact or less compactly packed. The cells present in cortex are thymocytes, epithelial cells, and the nerve cells. These thymocytes are numerous. Here in medulla, again, thymocytes are there, but these are few in number. Then epithelial cells are also present. Nerve cells are also there. And in addition, Hassel's corpuscles are also there. Their function is not very sure. Now, what is the process of development of T cells? We have discussed in the previous lecture that pre-T cells were formed in the bone marrow. Now, they will migrate to the thymus by the blood vessels located at the junction of medulla and cortex. This is the section of thymus gland. This is capsule. This is cortex. Again, we are dividing the cortex into two regions. That is outer cortex and inner cortex. And this is medulla. Now, the T cells. The pre-T cells will enter the thymus by the blood vessels located at corticomedullary junction. So here comes the pre-T cells. So the pre-T cells entering the thymus are also known as thymocytes. These are also known as precursor T cells. Now these pre-T cells will migrate to the peripheral or outer cortex and will undergo a series of stages which are known as DN1, DN2, DN3 and DN4. DN means double negative. There are two very important surface receptors on T cells which are glycoprotein in nature, that is CD4 and CD8. So both are absent in these type of cells. That's why these are CD4 negative and CD8 negative type of cells. But important thing is that although these are not having any active beta cell T cell receptor, but these are having invariant alpha chain, and there is also a beta chain locus. Now the DN1 will convert it to DN2, then DN3, then DN4. Finally DN4, in DN4 beta chain locus will rearrange and the alpha chain and beta chain will make a pair. So if pair is formed, that is beta chain locus is rearranged, then the pair is formed, and if this pair is formed, then two types of surface receptors will develop, that is CD4 and CD8. So as soon as the CD4 and CD8 both receptors are formed on the T cell, they will become double positive from double negative. Now they are having CD4 as well as CD8. Now this type of double positive cell, this stage, will migrate to the inner cortex, that is deeper part of cortex. This whole process which has occurred in the outer cortex is the first stage of development, which is known as beta selection. This is known as beta selection. Now this double positive stage will enter the deeper parts of cortex, that is inner cortex, and then one important change will occur, that is alpha chain is rearranged. Alpha chain locus is rearranged, and this alpha chain locus rearrangement will develop a capacity of antigens to interact with the self antigens present on MHC molecules. And if this chain is not rearranged, then such cells will die due to neglect. If pairing fails, then no signals will be sent in the periphery and the survival will not be possible in absence of sending the signals. Now the alpha chain locus is rearranged. Now these are capable of responding to the self antigens. Now in the inner cortex, the cortical, thymic, epithelial cells, that is CTACs, will interact with this DP stage, that is double positive stage. And if the antigens respond well to these, respond well to MHC class 2 proteins, then the CD4 will remain and CD8 will be lost. And if the interaction is in such a way that this will respond well to MHC class 1 proteins, then CD4 will be lost and CD8 will remain there. So now these stages have become single positive from double positive, and they have developed the interaction, the capacity of interaction with the self antigens present on the MHC molecules of the cortical thymic epithelial cells. Now these single positive cells will migrate to medulla, but before that we should know that all the events occurring in the inner cortex are termed as positive selection. Now why these are termed as positive selection? which cells are selected, which had developed the capacity of interaction. So they will proceed in the way. So they are selected to proceed. That's why it is positive selection. Now the single positive cells, these are single positive cells, they will migrate to the medulla. They are migrating to the medulla. Now they will interact with the medullary thymic epithelial cells. These are medullary thymic epithelial cells. Now this interaction with the self antigens on the MHC molecules of the MTEC cells, that is medullary thymic epithelial cells, will result in three fates. There will be three fates in case of both CD4 and CD8 cells. First fate is that they will respond with high intensity. That is, very strong interaction is there. Very strong interaction will be there. If very strong interaction is there, then they can produce autoimmunity also. So in this case, apoptotic signals are given to these cells and they will die. While some of the cells, this is the first fate, this is the second fate. Now some of the cells will develop in T regulatory cells. So this is the second fate. And the third fate is that those cells, those single positive cells which have given mild to moderate response or interaction are termed as naive T cells. These are immature T cells. And such cells, this is the third fate, which have given mild to moderate interactive response, are known as naive T cells or immature T cells. And these naive T cells or immature T cells, which is the third fate, will leave the thymus through the blood vessels present at the corticomedullary junction. Now in this step, these cells which have given very strong interactive response are selected to kill. So this is selection, but it is not to proceed them in the further developmental process, but to kill them. That's why the events occurring in the medulla are termed as negative selection. Various hormones play important role in these maturation process or developmental process, like thymolin, thymopoietin and thymosin. So this is about the development of T cells in thymus. Hit the like button, share and subscribe our channel to get the latest updates and notifications.